It's great to be here and participate in this super interesting talks and discussions. So I'm going to talk about how we developed a stem cell-based therapy for Parkinson's disease. And I'm sure that you all know the disease. The rationale for using cell-based therapies in Parkinson's is really two. The first one is that the core pathology is loss of the midbrain dopamine neurons. So it's one type of neuron, and they do one job, and they release dopamine in the striatum, and there they modulate movements. So what you need to replace is one cell in one location. So this is really the easiest way when you go into the brain. And we also know from patients that if you replace the dopamine levels, the patients do very well. So you give them L-DOPA, you increase the dopamine levels in the forebrain, and you have good effect for many, many years. But when you deliver L-DOPA or similar medications, it's associated with a lot of complications over long-term use. And this is because you get a global delivery and an unregulated release. So cell therapy is thought to be better because you replace dopamine only where it's needed, and that could then reduce the off-target effects. You release dopamine in a physiological way, and you would avoid the long-term complications with L-DOPA therapies. So this was really pioneered at Lund University, where I work, more than 30 years ago, where Anders Björklund and Ola Lindvall dissected out pieces of the fetal midbrain. So this is the midbrain from aborted embryos, made a cell suspension, transplanted into the striatum of PD patients. And from these trials, and there's been a couple of hundred patients grafted globally since then, there's been a highly variable outcome, but some of the patients have had a long-term clinical benefits. And Paolo Piccini here in London has shown that the dopamine is released from these cells in a physiological fashion, and the cells survive for more than 24 years. And here you see a graft from patient number 3 in Lund, and this is a 24-year-old graft. So the graft core is here, the cells are healthy, viable, and functioning, and the lighter gold here is the innervation into the host brain. So really it can work very well, but there's a huge variation. So a while ago there's this initiative TransEuro that materialized into a new clinical trial using fetal cells. So it's more or less the same thing under slightly more standardized conditions, and primarily there's a better patient selection in this trial. However, we did graft the last TransEuro patients. Cambridge finished last year, we finished this year. But no matter how good the outcome of TransEuro is, fetal tissue is not a way forward for treating a large number of patients. It's very low ability of the tissue. For every transplant we performed, we canceled 17 for lack of tissue. It's not possible to standardize or QC the cells before the testing, and we think this is enlarged explains the high variability in the patients. And there's a lot of ethical and societal barriers, at least in some regions of the world, where fetal tissue is not a very good source to use. So for a long time, my group has been working on developing alternative sources of cells, and where we're closest to the clinic now is stem cell-based therapy, and we start off using human embryonic stem cells. And this is work that's been progressed over many years, where you start with developing a good differentiation protocol, assessing the function and authenticity of the cells that you generate, and then we moved into establishing a GMP manufacturing process, and now we're preparing for the clinical trial, which will be a small open label trial. So I'm going to talk today about how we use different stem cell lines and ensure reproducibility in some of the steps where that's been a key issue. So developing a good dopamine neuron differentiation protocol doesn't sound like a big feat today maybe, but if you go back to 2010 when we started this, cells could be differentiated into dopamine neurons, and there's a number of different protocols for that, but they didn't really have a midbrain identity, they didn't survive very well after transplantation, and if they survived, they didn't really function, and there was lots of problems with uncontrolled proliferation and tumor formation. So when we sectioned these brains, big tumors just fell out. So we developed a protocol based on the dual SMAD inhibition from Lawrence Studer's lab, where we use extrinsic patterning agents to set the rostrocaudal and dorsiventral level of the cells. And we can make forebrain, midbrain, hindbrain, and here you see VMs, so ventral midbrain progenitors derived from this protocol. And when you transplant these cells, in the first study we used up to 18 weeks. This is as far as you can go in the cyclosporine model, which is the most common xenograft model. But we've since done a lot of studies in nude rats up to 12 months. You see the transplant here, it's the lesion side. So this is the intact side, this is the lesion side. The transplant survived well, innervate the host targets. And when we do our functional assessments, whether it's drug-induced rotation tests or spontaneous motor tests, we could see a full behavioral recovery in these animals. When we developed this protocol on H9 from YCell, when we published it, we did reproduce the key findings using a cell line from CellArtist in Sweden. And the reason was that was really an editorial requirement, because I think at the time, H9 had the rep to be the only cell line that was good at generating neurons. So if you made neurons from H9, they weren't so impressed. So at the time, we did replicate this. And then, of course, our question was, are the cells good enough to use in patients? Well, it's actually very, very difficult to answer that question, and I think the discussions today have highlighted that it's, there's not really a good answer. But we really answered it by asking another question, and that was if the stem cell-derived dopamine neurons are equivalent to the fetal dopamine neurons in the developing human brain. And luckily, we have access to this fetal tissue in Lund, so we could do a lot of direct comparison studies in our long-term xenograft models. I'm not going to go through any of this data today, it's all published, but basically we could show that stem cell-derived dopamine neurons function equally well and in the same way as fetal dopamine neurons. And this is in terms of their survival, potency, phenotypic maturation, how well they restore motor deficits, how they integrate into the host brain, and how they innervate the correct target structures. So all this data tells us that yes, they do behave very similar to the fetal dopamine neurons. So that answers the question, yes, they're ready to go to clinical trial. During this work, we switched a lot from H9 to RC17, sometimes keeping them in parallel. RC17 is a GMP line from Roslin cells. There wasn't really any particular reason for this, except that we were part of an EU project, and Roslin cells was part of that project. And as part of the EU project, we tested the lines. RC17 worked really well. But basically, most of these studies have been replicated in H9 and RC17. And this turned out to be good, because going to clinical trials, we've been, and Pete has been involved in some of these discussions, we've been going, should we go with H9, should we go with RC17, H9? Both of them would be possible to use for the first clinical trials. So we've been moving back and forth a bit. When we did the GMP adaptation of the protocol, we used RC17. And I'm not going to go through all these steps, but really in 2012, when we published the differentiation protocol, it was fully defined and xeno-free. And at that time, I thought that meant, well, it's basically GMP, isn't it? Well, it turns out that it's not. We changed every single component in that differentiation. And GMP is also a lot about reducing the variability. And I'm not going to go into the steps, but it took us many years. But we were actually quite lucky in this process too, because one of the key things that we needed to change was the matrix that we grow the cells on. And our research grade protocol had a free-floating EB, or today you probably call it an organoid stage, but a free-floating stage, which was hard to reproduce in the GMP facility. So we tested different substrates and ended up with laminin 111. So this was really just to get the cells to attach. But luckily, it also increased the yield more than 40-fold. And you see that I have upscaling in brackets here, because we did have a lot of contacts with companies to do automated manufacturing and upscaling. But it turns out that with the yield in our GMP protocol, we can do the first batch of cells just using manual manufacturing. So starting from 1 6-well plate of ESL, so starting from 12 million cells, we will at day 16, which is at the day that we transplant the cells, we can make 500 doses for patients. And at peak production, we have 14 flasks. So this is super efficient and super quick. And the quicker it is, the less regulatory issues there is to deal with. So we haven't actually upscaled this process. We did test, and this was that the GMP differentiation protocol worked on a number of lines. We tried the RC17 and the H9. We also tried cell lines from Karolinska Institute and some of the Sheffield cell lines. And as you see, here you see 4 of these lines. We've tested many more. They do respond very similar to the patterning factors. You might have to change the concentrations a little bit, but in large, these cell lines all differentiated very well. The reason that we tested so many lines is in part because when you do go to the patients, it's not just about what is the best cell in terms of what is the best cell that can make the cell generate the neurons that we need. You have to look at the ethics, how was a consent form from the donors acquired, what's in there, what year was it derived, because depending on if it was before a certain date or after a certain date, there's different regulatory requirements. The global compliance, the lines that exist today. So the RC17, which is a European line, is not possible to use in the US at the moment. And for commercial aspects, there's a lot of issues with what cell line to use, and we might get back to this in the panel discussion. But it's good, I think, to make sure that your differentiation protocol works on a large number of lines, because if you lock to one line, you have no possibility of switching lines when something comes up that warrants that you would need to do this. Now, we also developed a cryopreservation protocol, and in this work, I just show some of the data here. We have the RC17 fresh and frozen. We have HS980, which is another Karolinska line. So we take multiple lines to test every step. And here you see the behavioral recovery, where we, again, went back to H9 to make sure that that line still works, and some of the other lines that we work with. Now then, once everything was established, and again, this was just to have flexibility in choosing a line, or if a better line came up, to use that one. Then we were supposed to produce the cells at Roslin Cells, so they did 3 GMP 1/6 of the scale runs. And that was, of course, only done with RC17. And we could show that with our GMP protocol, we got behavioral recovery from all these 3 GMP batches made at the GMP lab as well. Now, for a number of reasons that I don't really have time to go into, we had to switch the facility. So now we're producing the cells at Royal Free Hospital here in London. And I must say that has been the smoothest technology transfer I've ever done, both because they're super professional, but also we know exactly that each step of the process is very robust. So to switch this facility wasn't as bad as it sounds. So we're back on track. We had our meeting with MHRA 2 weeks ago, where they basically, we suggested how so the way these regulatory meetings work is we suggest how we want to test the cells in all sorts of ways, and then they approve or make comments or make suggestions. And I think thanks to Pete, we've had a breeze. We've been flying through this process. And a lot of the issues I know that you've had to deal with, they're now familiar with the site of transplantation. It makes more sense to put it in the organ of where you need the cells, etc. So in terms of genetic stability, for our product, they will require karyotype, and make sure that there's none of the oncogenes that are mutated. And then we're going to look at the PD risk variants, because of course, if you have a stem cell line for PD, you don't want to have a donor that has one of the PD risk genes. So this is all that they require, and this is all that we will do. Now, that might seem worrisome to some of you, but you have to think that in this setup, we have this final cell product. We will make 300 vials. 50 of those will go for QC. 30 will be our backup reference vials. And then we will have vials for safety and efficacy and 150 vials left. So that's enough for 75 patients. So the exact same cells that will go into the patients, they will also have been going through this QC. So in addition to this, we will, of course, on the same cells then, do safety, which is histopathology, biodistribution, and tumorigenicity. So we will know in functional assays that the cells are safe and that they don't venture outside of the brain, etc. And on the same cells, we will also do efficacy testing. So these tests have now all been okayed by the UK authorities. That's going to do the voluntary harmonization process with the Swedish authorities. That's going into this for the first time. So we're hoping that it will be that it will be smooth as well. And this means that the cells are being produced right now, so we will have them before Christmas, and then we need about a year to do all the safety and efficacy testing on that batch, ready to go into patients a year after we get the cells. Now, what will this clinical trial look like? And I don't have a lot of time to talk about this, but we've done the transuria trial in Cambridge and Lund. It's headed by Roger Barker from Cambridge. And in this transuria trial, we have a large cohort of patients that go in the observational group. And from this observational group, where we can look at the natural progression of the disease, we have the transplant group and the PET controls. So these are the transuria patients. They get followed up PET scans every 6 months. We transplant, so the endpoint is 3 years after transplantation, which is after the last patient was transplanted, which was March 1st this year. So in 3 years we will know, and then we'll continue to assess them long term. And then we have the clinical team in Lund here. These patients are both in Lund and Cambridge. So the plan is to draft the STEM-PD, or the stem cell patients, from the same observational cohort, which is then already established and followed for 5 to 10 years at these clinical sites. And then we will recruit 12 patients to our stem cell project, half of them to control and half to receive the stem cells. And then in this remaining control cohort, there's also, that doesn't exclude the patients from doing anything. So some of the patients have developed so far in the disease, they've gone into advanced treatments. So we have DBS patients, etc., and duodopa patients in here. So really, we will be able to compare fetal VM tissue, stem cell dopamine neurons, and best available therapies from the same patient cohort. So I think this is a really nice setup for the clinical trial. So my last slide, I just want to end with saying that this is really a global effort to bring these stem cell-derived derivatives to the patients. We have established a network called GeForce PD, where we work together with American teams and the Japanese teams working on hESC and hPSC therapies for Parkinson's disease. It's been extremely helpful, both when it came to developing the cells, but also to deal with the regulatory authorities. And the work here in Europe has been performed within EU networks, and I was lucky to, crash the UK regenerative medicine platform, because we don't really have that kind of structure in Sweden. So I benefited immensely from being able to be an associate partner, or whatever I was, in this network that really helped us to understand all the questions that was relevant for bringing the cells to the clinic. And here's the core team. It's Agnete Kirkeby that's been working with me for many years, heading the GMP protocol development. That's me. Roger Barker, that's going to lead the clinical trials, and Danielle Daft, that's managing everything from Cambridge. So thank you.
So any questions for Malin?
It's a statement rather. You know your 30 reference files, will some of those be for long-term stability testing?
Yeah. So we will need, so this is something that the authorities are going to get back at us, whether we can do this long-term stability testing on the test batches or if we have to do it on the real batch. We have done stability testing, so we've had cells in the freezer in my lab for 2 years and then transplanted them and they're fine. But it's not trivial how, oh, you're getting a phone call. Yep. It's not trivial how to assure, and the methods you usually do for drug testing, where you store them at suboptimal temperatures, for example, to accelerate, mimic an acceleration of the time in the freezer, can't really do with the cell product because it just kills the cells. You know, it's not like the effect will slide, it will just kill the cells. So probably we'll do it both with the test batch and with the real batch. And then we want to extend, extrapolate from the test GMP batch.
So presumably there was never any QA or QC on the fetal cells even retrospectively?
Well, it's not really. So this has been taken through all the authorities in Sweden and the UK for the Transeurope trial.
Yeah.
There's a few tests done on the donor of the,
The mother.
well, the donor, she's not a mother yet, I guess. And then that's HIV and syphilis and basic things. And then we collect the cells and we can keep them for maximum 4 days. We need tissue from 3 embryos. We have just enough cells to do one thing, and that's we do a trifin blue dye exclusion method.
Wow.
Right before taking them into the patients. And, you know, the reason that was approved is that that's the best you can do. It's not good.
No whole genome sequencing.
No, you know, you have no time, you have no tissue. So this has been problematic.
Yeah.
Can I just ask how you settled on a cell dose? Thank you. How you settled? I didn't quite catch it on your slide.
Yeah.
Have you got a dose ranging study as part of this, or?
So the cell dose will be based on the transplants that was done in the late 80s, early 90s. If you look at those patients, they have a good effect, need about 100,000 surviving dopamine neurons. So we can take our cells. So one thing we've done is to ensure that they work at equal potency to the fetal cells. And then we can just in our animal models calculate if you transplant 100,000 cells, how many surviving dopamine neurons do we have after 6 months? Then we can calculate the dose. The dose escalation study will start with 75,000 cells because we expect to get some efficacy. And then 6 months after, we will decide if the second group of patients will get the same dose or a higher dose, and that will be based on the PET scan because you can't really see any effect. The complication is that you transplant the cells when they're immature progenitors, 16 days after being pluripotent, and then it takes them at least 6 months to start functioning and 3 years before they're fully functioning. So it's all, you know, it's all hard to do dose finding studies under those circumstances.
The earlier fetal studies, though, my understanding was there wasn't a very good correlation between cell survival and cell dose. And of course, it was difficult, I realized, to get the cell dose consistent with those earlier studies. But it implies from what you've said that the correlation was strong enough for it to be predictive for this study. Is that?
Well, the number of surviving dopamine neurons?
Yeah.
Yeah, I mean, basically, if the cells didn't survive, the patients didn't have an effect. Yeah. Patients that had good cell survival tended to have a good effect, except that there is one patient from Jeff Cordover's trial in the US that they came to postmortem, because most of them, it's hard to get the brains back after 25 years. But that patient had no effect of the transplant, and when the brain came to postmortem, you just expected there would be no cells. But it looked good. So maybe there's one exception to the rule. We don't know, but a general consensus is that you need to dose around 100,000 surviving dopamine neurons.
Yeah, and following up on that, on the slide you showed with the GMP protocol, there were around 5,000 TH positive cells on average at would, or is it 5,000?
That's per 100,000 cells grafted.
So 5,000 per 100,000, or 50?
That's the average.
Yeah.
But we will set that dose exactly for the clinical product.
And was that 5,000 cells that were all of them TH, or the 95,000 cells were something else?
So in the fetal grafts, about 5% are dopamine progenitors, and in the grafts, the therapeutic neurons is a minor component. For the ESLs, we know that the dopamine neurons is a higher proportion. But up until now, we haven't known actually what's in the graft except dopamine neurons. And this is a question that's easy to ask, extremely difficult to address. It's been a question in the field since the fetal cell transplants in the late 80s. Because of course, right now, we're banking on the fact that they're the same. But we're now actually looking into whether it is, what are the other cells. So the authorities, as long as you characterize what's in there, they don't make it sure it's not harmful, but for scientific purposes, of course, we need to know this.